when we change the sequence of DNA in such a way that the sequence in the mRNA, even after it's edited, is changed. And that causes some problem with that individual. It causes some genetic defect. It causes some disease. Then we call those mutations. So in general, we tend to use the term gene polymorphism to describe mutations that are silent, that don't have any deleterious effect, that don't hurt anybody. So if it's just a difference between me and you probably know already that me and you are genetically different, we're not identical twins, and so we have genetic differences, but we code for the same sequence of keratin, for example, in our hair, but it just has a slightly different gene sequence. We call those gene polymorphisms. But if it caused me to code for the protein of keratin that makes up hair that was defective in some way so that I had no hair, a condition called alopecia, then we call that a mutation. So the difference between a gene polymorphism and a gene mutation is generally that the gene polymorphism does not cause disease. It's just the difference between different individuals, where a mutation does cause a disease, or at the very least a noticeable change in the organism, like the amber, umber, and ochre eyes that we saw in the fruit fly. The abnormally colored eyes of the fruit fly would be examples of mutations in fruit fly. So we have different types of mutations. Remember that we have a reading frame. We read the codons in groups of 3 bases, CAT, 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 CAT. If we have a point mutation, it doesn't change the reading frame. That reading frame is that group of 3 letters, CAT, CAT, CAT. If we have a point mutation, for example, we change one of those T's to an A, it doesn't change the reading frame. It's just CAA, CAT, CAT, CAT. If they don't change the protein sequence, in other words, if we go back to the codon table and we see that CAA and CAT are codons which code for the same amino acid, then how will we ever know that that mutation took place? Because it's going to produce the same protein with the same amino acid sequence. So we call that a silent mutation. But if they do change the protein sequence, again in a way that doesn't change the reading frame, just a single nucleic acid substitution that changes the protein sequence by changing where we are in the codon table, then we can call these missense mutations if they change one amino acid to another, or nonsense mutations if they change a codon which codes for an amino acid to a stop codon, which is going to result in an abnormally short protein. We can also have frame shift mutations which change the reading frame. If I insert a letter between T and C, now instead of CAT, 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 as I'm reading along, I have CAT, ACA, TCA, TCA, TCA. I've changed the reading frame. It's one of these missense mutations which results in the disease sickle cell anemia. Once again, we're back to our familiar beta globin gene. Poor beta globin gene, it always gets hated on. There's always problems with the beta globin gene, as though this is the only gene that gets mutated. But we keep coming back to examples of the same thing over and over again. Hopefully you'll become familiar with beta globin and feel comfortable with all these different examples of all the hideous things that you can do to your beta globin genes. In this particular example, we're taking that beta globin gene. It's got a sequence in DNA that's CTT, and that codes for, eventually, the amino acid glutamate. Remember, glutamate is an acidic amino acid, so it's polar, it's highly charged, it likes being in a charged, water-filled environment. Here we have a mutation in the beta globin gene, that changes the codon GAA on the mRNA, which codes for glutamic acid or glutamate. Remember that glutamate or glutamic acid is a charged acidic amino acid, likes hanging out in a watery environment. We change that GAA codon to a GUA. We change the middle letter from an A to a U. This codon, GUA, codes for the amino acid valine. Valine, it turns out, is a nonpolar amino acid. So we've gone from an acidic polar amino acid to a nonpolar amino acid. We've gone from a hydrophilic amino acid to a hydrophobic amino acid simply by changing one letter in one codon in a whole big string of letters that are found in this mRNA. That completely changes the structure of the hemoglobin molecules, which completely changes the structure of the red blood cells. Now the mutation that's shown here, if you're really sharp-eyed, you'll notice that this is a slightly different mutation, but it still produces the same result, which is to turn a glutamic acid into a valine. And it turns out that the glutamic acid, which lies at that point in the protein, primary sequence, is essential for the proper folding of that protein into a secondary sequence, into a tertiary sequence, and into a quaternary sequence. So by simply disturbing the primary sequence of the protein with this mutation, we've disturbed the secondary, the tertiary, and the quaternary structure of this protein in such a way that instead of folding hemoglobin up into nice balls of yarn, we end up with rigid rods. Because where there was glutamate that liked being around water, now there's valine that hates being around water, and that ends up changing the structure of the protein. And that structural change results in the disease sickle cell anemia. Well, here's an argument for human evolution. Sickling's bad for you. Having your red blood cells collapse in on themselves and form these sickle shapes is not a good thing to have happen. Why is it that people with this sickle cell mutation don't die before they can reproduce? Or more to the point, why is it that they're so sickly? Get how that works? Why isn't it that they're so sickly, they're so sad, that they can't find mates, and so they have a lower reproductive fitness? We would imagine that having a sickle cell mutation, even if it doesn't kill you, would lower your reproductive fitness. And so that should eventually eliminate this mutation from the population by the theory that we learned, the theory of natural selection. It turns out that there's a counterbalancing factor. The sickling protects red blood cells against infection by the malaria parasite. In Module 6, in the next module, we'll look at how it is that red blood cells are infected by the malaria parasite. But at this point, all we need to know is that red blood cells are infected by the malaria parasite. And we interfere with this infection process if we have sickle-shaped cells. So now we have a balance. The people with sickle-shaped red blood cells don't run, jump, and play as well as the people with normal biconcave disc red blood cells. But the people with normal red blood cells, that is a biconcave disc, have a higher incidence of malaria, so they don't run, jump, and play very well either. So now these two things balance themselves out. And, as we might expect from this model, the distribution of sickle cell mutation in the population is a good match to the distribution of where there's malaria, wherever there's these balancing forces. Everywhere else, because malaria wasn't a problem, the people with sickle cell did eventually get eliminated from the population. But where there's malaria, where there's this counterbalancing factor, then there's more sickle cell anemia. So this is an argument for natural selection in primordial human populations, which is an argument for human evolution over hundreds of thousands or millions of years. Well, I mentioned earlier frame shift mutations. These are the mutations which change the reading frame. Recall that we're reading 3 bases at a time, CAT, 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 CAT. In deletion mutations, which shift the frame, 1 base is removed, shown by a vertical red bar here, CAT, CTC, ATC, ATC, ATC. See how that changes the sense by changing my reading frame? Or if I have an insertion mutation, I add a base, let's say the base C, shown here in red, then I read along in groups of 3, codons CAT, now I have CCA, TCA, TCA, TCA. That's changed the sense of what I'm reading. These are both frame shift mutations. One's a deletion, one's an insertion. Both have changed the reading frame. Both types change the reading frame.